Assalamu alaikum to everyone present here today. Welcome to everyone to this research webinar, the series 2.0. My name is Jamal Hosseini, the moderator for this session. So this session is going to be about 1 hour 15 minutes, 1 hour of the presentation by Dr. Hio, and about 15 minutes of a Q&A at the end of the session.
Sure.
If should anybody have any burning questions, you can please put it up in the chat, or you can bring it up at the end of this session. So today we have my very good friend, Dr. Hio Chong will be discussing, or will be rather sharing his experience searching for dead penguin in Antarctica. Very interesting title. Before I give you the floor, Dr. Hio, I'd like to give you a quick background about you to the rest of our audience. We're having 120 and counting audience today. So let me give you a brief description or background of Dr. Hio. Actually, it's very short one, but there's a lot much more about this to talk about Dr. Hio. Dr. Hio Chong obtained his Bachelor in Medical Sciences by the honors and Masters in Medical Sciences Parasitology from the University Kebangsaan Malaysia. He then earned his PhD from the Department of Entomology, Texas A&M University in 2016. He is currently a faculty member at the Faculty of Medicine, University Technology, MARA, UIT in Malaysia. Prior to that, he was a research officer at the Entomology Unit, Institute for Medical Research, Kuala Lumpur. His research interests are carrion decomposition ecology, biodiversity, necrophagus, and coprophagus dipatron species, and forensic entomology and aquariology. He has received the Natchatram Medal 2018 from Malaysian Society of Parasitology and Tropical Medicine, the John Henry Comstock Graduate Student Award from Entomological Society of America during the annual meeting at Denver, Colorado 2017. He's also a recipient of the Perdana Scholar Award from the Malaysian Embassy at Washington, D.C. 2016. Additionally, Dr. Chen is a board-certified entomologist since 2016. To date, he has published 55 peer-reviewed journal articles, 1 book, 2 book chapters, and 35 articles in magazines. Besides, he has 6 patents and more than 20 copyrights submitted to the Intellectual Property Corporation of Malaysia. Currently, he is a member of the Malaysian Society of Parasitology and Tropical Medicine, associate member of the North American Forensic Entomology Association, and associate member of the European Association for Forensic Entomology, and associate fellow of iPerform UITM. With this brief introduction, I will hand over the floor to Dr. Hio to share his experience with us on searching for dead penguins in Antarctica. We are all eagerly waiting. Dr. Hio, the floor is yours.
Yeah, certainly, Dr. Jamal. Thank you very much for the nice introduction. Yeah, and good morning, salam sejahtera to everyone here. I'm very happy to share my experience with you all. I would like to present, share my screen with you all now. Let's see whether it can work. All right. All right. Can everybody see the slide?
Yes, we can.
All right. Okay. So just let's get it started. So yeah, this is the picture that I put for my poster. But as you can see, I did not wear the face mask properly. I think because this is way before the pandemic. So not many people care about how to wear the mask properly. Yeah. All right. So the title of my presentation today is Searching for Dead Penguins in Antarctica, my research experience. And as Dr. Jamal had mentioned just now, that I'm an associate fellow from Institute for Pathology, Laboratory, and Forensic Medicine, iPerform. from Faculty of Medicine, UITM, Sungai Buloh campus. And I will start with my presentation with the introduction. I would like to talk briefly about my background as well as I perform. And then I will enter our topic today, which is the necrobiome study on penguin carcasses. And at the end of the presentation, I would like to share like how we are going to get this grant so that we can work in Antarctica. Sorry. Yeah. So let's start with the introduction. So I perform stands for Institute of Pathology, Laboratory and Forensic Medicine, where Dr. Jama is a fellow and myself is an associate fellow. And the niche area of this institute are actually diseases such as atherosclerosis, metabolic diseases, epidemiology, natural products, as well as forensic science and medicine. And this is how I can fit into this niche of this institute, because I myself is trained as a forensic entomologist. And this is the organization chart of iPerform, and you can have a look from our website, the iPerform website, as well as the iPerform Facebook page. And a little bit about the history. I joined iPerform as an associate fellow back in 2017, and at the time we have conducted this forensic entomology workshop, where we have attracted around 20 to 30 participants from many universities to learn about what is forensic entomology. As well as last year, iPerform also co-organized a seminar with Malaysian Society of Palastology and Tropical Medicine. And the title of the seminar is "What Else Can Fly Tell About Death?" So we have 5 speakers, 2 from US, 1 from Thailand, and 2 from Malaysia. And one of our speakers is actually a police officer from PDRM. And also last year, at the end of last year, December, we had one press conference before my departure to Antarctica. So personally, I like this quote a lot. And who saw him die? I set the fly with my little eye. I saw him die. Meaning that flies, they are everywhere, and they are the best witness. When there is a murder or crime happen, perhaps flies can tell us who is the murderer, if we understand the behavior, the ecology of flies. Yeah, they are the good witness, actually. So I took this picture myself. This is called a hairy maggot blowfly, Chrysomya rufifacius, in Selangor. Yeah. So allow me to introduce a little bit about what is carrion ecology before we enter our topic today, because I think it's a good background for everybody to know why is it important to study carrion decomposition, yeah, the dead body decomposition. So what I mean carrion here is dead and decaying carcasses, yeah? Animal carcasses or dead bodies, right? And traditionally, people don't really pay a lot of attention on animal dead bodies or human corpses. Yeah, because the biomass on Earth, actually majority, I would say like 95%, from plant. Yeah, the biomass of plant actually dominated the Earth. When they decompose, the plant decompose, they really give back a lot of nutrient back to the ecosystem. So only 5%, are contributed by animal bodies or carcasses, right? However, recently, carrion has been attention, because when carrion decompose, or when a dead body decompose, it actually changed the soil biogeochemistry, yeah, underneath the soil, as well as the invertebrate communities in the soil, right? So carrion actually offers significant ecosystem services, right, to the ecosystem. And what we are interested as a carrion ecologist is we would like to explore the mechanism of species interaction, because we know in the soil there are many other arthropods like mites, insect larvae, beetle. So we would like to know what is the interaction among them and how they can work together to decompose the body, right? And we know that carcasses do not decompose in a vacuum, right? actually they do it so in an ecosystem where all the members, the players in the ecosystem work together, like the bacteria, like the arthropods, like the worms, the mites, they work together to compose it, right? And from there, they have some evolutionary pressure, and then so that they can more well adapted to the changing environment. So this is the Carrion Food Web. As you can see, there, the dead body is in the middle, right? And who are the consumers for carrion? Who consume dead bodies? Yeah, of course, first and foremost is microbes and fungi. They are really good consumers of carrion. Yeah, I would say they are the first to start to decompose carrion. Then followed by the predators and scavengers, and this can be like the wildcats, the lizards, bear. Those are scavengers, as well as detritivores. And here means that those are insects like flies, they lay eggs, and then the maggots feed on the carrion. So those are the consumers. Also, carrion can contribute substantially to the soil nutrient. And this is very important. Yeah, carrion, when they decompose, then return nutrient back into the ecosystem. So your soil is fertile, and then you can plant. Your plant can grow from in the soil due to the nutrients. And here are the application of carrion decomposition. This is why we need to study carrion decomposition. First is to maintenance of hygiene and public health standards. And this is very important. Can you imagine if dead bodies or animal carcasses cannot decompose and they stay in the environment forever? Probably we are working on the dead bodies right now because too many of them and cannot decompose. And when they cannot decompose, bacteria will proliferate on the carcasses and then disseminate to the environment. And this could cause disease outbreak. That's why carcass, they must go, right? Carrion, they must go. Yeah, that's why it's important. Second, transforming food organic waste into proteins. And this is a trend where people use maggots to recycle food waste or organic waste in the dumping ground. Yeah. Let's say you have a poultry farm and you have chicken, and chicken poops is a big problem, right? So now a lot of farmers, they use maggots to feed on the chicken poops. They clean up all the chicken feces. And then when the maggots get bigger, they actually fed the chicken with the maggots. So it's kind of you just recycle the maggots, right? Yeah. So this is insect protein. Number 3 is biodiversity conservation. Actually, believe it or not, a lot of animals, like scavengers, that depends on carrion or carcasses as their food source. Without carrion, a lot of animals, they cannot survive because there's no food. Yeah. But today, our topic is to focus on forensic entomology. Yeah. When the body decomposes, is actually valuable in forensic investigation. And I would like to talk more about this. So what is forensic entomology? It's also known as medical legal entomology. It's a use of insect evidence to answer questions pertaining to legal issues. Yeah, and in Malaysia, we are using forensic entomology as evidence as well in the court. And forensic entomology includes arthropods' involvement in the events such as murder, suicide, rape, abuse, trafficking, as well as movement of body. Yeah, and there are many applications of forensic entomology as well. Yeah, like the first one, determination of time of colonization or minimum post-mortem interval, is kind of a technical term. Actually, I can put it in a simple way, it's a determination of time of death. Time of death. Because we know that when a person or an animal, then they die, as soon as possible, the flies will come and lay eggs on the dead body, and then the eggs hatch and maggots emerge, and then the maggots feed on the body. So if you can estimate the size and the developmental stages of the maggots, you can actually estimate the time of death of that particular disease or victim. Yeah, so that is the most important, the major usage of forensic entomology. Of course, forensic entomology can be used in determination of cause of death, location of death, body relocation or body movement, site of injuries, torture, and many other. And each of these applications, you need a lot of research to back up, a lot of scientific evidence, scientific methods to prove it in order to be presented to the court. So this is what happens soon after death. Fly arrive at the corpse as soon as possible, usually within 10 minutes. So this is what I'm saying here is usually under normal weather condition, under normal circumstances, which means there's a warm, outside, there's no rain, daytime, then flies usually will arrive as soon as possible. And then the flies start laying eggs or larvae in the orifices or wounds, like mouth, ear, eye, or of the dead person. And then the larvae, which is maggots, consume flesh of dead body and go through the life cycle, right? And this is the life cycle of blowfly. For example, the adult fly lay eggs. You have fly eggs. In one day, the egg hatches, and then the fly larva emerge. And then the fly larva feed on the body. About 7 days, they've transformed into pupa, right? And then pupae, from pupa, the adult fly emerge from the pupa. So altogether in Malaysia, depending on species and temperature, about 10 days, or sometimes less than 10 days, for the complete life cycle from eggs to adult. All right, and this kind of life cycle, we call it a complete life cycle, right, or holometabolous. I would like to give you an example. It's quite interesting. It's a book entitled "Washing Away of Wrongs." It was published in 13th century in China by Zhongzi. It was a real documentation about how the ancient forensic investigator used flies to solve their crimes. So the stories goes like this. In 13th century of China, a farmer was killed with multiple slash wounds in a paddy farm. So the Chinese investigator Zhongzi was called to investigate this criminal case. Right, as you can see in the picture, there's the investigator, and he noticed that the wounds were injured by a seeker, as you can see in the picture. Yeah, so he asked all the village men to assemble, each with their own seeker, because he said he knows who is the murderer. Just show your seeker to me. So in the hot summer sun, flies were attracted to only one seeker because of the blood stain or the smell on it. And finally, the owner of the seeker confessed to the crime. And this was a story presented in the record. Yeah. But of course, nowadays we need a lot of scientific evidence and scientific foundation to prove that, in order to use in the court as well as our services. So for your information, we do have forensic entomological services at UiTM Sungai Buloh. I just want to share with you my personal case, a real case back in 2017. It's also my first case after I done with my PhD. So this is a picture that we took, and behind the scene was a body recovery site. So a body of unknown female with multiple blunt trauma was discovered in May 2017 in Selangor. The body was partially buried with the skull exposed. Yeah? The skull was exposed on the soil surface. So during autopsy, 9 insect larvae were collected from the skull interior, inside the skull. Yeah, they found 9 insect larvae. The pathologists found and collected. Here is the picture of the 9 insect larvae. And what interesting about these maggots are, they have tails. Can you see the tails? It's very peculiar or weird looking maggots. So actually, we have a common name for this species, this kind of organism. We call them rat-tail maggots because of the tail looks like a rat tail. The tail is actually for swimming, for your information, and the tail is like a telescopic tube in order to get oxygen, meaning that these larvae are living in the aquatic, in the water environment, but the tail is going up to get the oxygen. It's just like the scuba diving. They have a pipe, something like that. Yeah, this is the close-up view of the maggot. So as all the larvae collected from the skull were dead, there were no larvae available for rearing purposes. So we actually need live maggots. Yeah, we need that as well as the live maggots, because sometimes it's very difficult for us to identify the species of the fly based on the dead maggot. Sometimes we need live maggots so that we can rear them into adult stage. We need to see the adult, what kind of adult fly, so that we can identify it better. Yeah, but to solve this problem, a visit to the body recovery site was conducted after a week after the autopsy. Yeah, we put up the fresh cattle liver bed right nearby the body recovery site, in hoping that we can attract the adult flies to lay eggs, and then we get a larva. We need a live larva, so that's why we went back to the crime scene, the death scene. So we placed the cattle liver in a plastic container filled with rainwater just to imitate the natural oviposition site, the natural egg laying site. So this is what we have done. We actually make 2 holes on the plastic container just to imitate the eye socket. We believe that the flies, because the skull was exposed on the ground, so we believe that the fly actually go in the eye socket and lay eggs. So we actually make 2 holes on the plastic container. Hopefully the flies will also go in and lay eggs. Unfortunately, no shearfish larvae were found in the liver bed after 5 days. Yeah, we went back and checked after 5 days, no larvae. But we were fortunate enough that we actually discovered 9 live maggots moving in the trapped water of the barrier site. Just at the original site, the water was still there, and we found another 9 live maggots. So we were happy, and we brought them back to our lab, and we reared them to the adult stage. So we finally got the adult, and the fly looks like this. Yeah, it looks like a bee, but they are not bee. They are actually fly. Yeah, we also call them a flower fly. So after we worked with our collaborator, we identified this we recorded this as a first time as a forensically important species. But unfortunately, we do not have the data, the local data specific to this species, particularly in the developmental stage and duration. So therefore, the time of death could not be determined because lack of local developmental data. Although in Europe, they have done such study, but because in Europe, the weather is different, temperature is different, therefore we cannot use the data in Malaysia. We can only use it as a reference, but we cannot really determine the time. But because of the interesting discovery, we have published it as a record in Journal of Forensic Science. Yeah, a new species of forensic importance. Now I think now you all are more understand like what is my background and why am I doing this. Now we know that decomposition studies have been conducted in tropical and temperate zones, but what about polar regions? Right? In other words, how do penguin carcasses decompose in Antarctica? I think this is a very interesting question, very fundamental, but not many people knows the answer. Yeah. So this is the question that I posted in my proposal that I submitted to Sultan Mizan Antarctic Research Foundation. Yeah. You have to ask a good question, an interesting question that catch the eyes of the reviewers. Yeah, how do penguin carcasses decompose? This sounds simple, but nobody knows about it. Okay, no, the following slides is all pictures, and I just like to share with you my journey, like how I put myself there, right, on this research project. Necrobiome on penguin carcasses. Necrobiome. Necro means dead, anything dead. A biome is all the genetic material associated with the dead bodies, like the bacteria DNA, the arthropods DNA. So we plan to use like next generation sequencing, right, to do the metagenomic studies to identify what kind of bacteria are there, right? And here's our team members. We have 5 team members all together. Myself is the PI, and Dr. Jama. Yeah, Dr. Jama, are you here? Yeah, Dr. Jama is a fellow of iPerform, also is a core researcher in this proposal, as well as Dr. Siti Sofo Ismail from UMT, Terengganu, Dr. Lucas Low from UM, and Mr. Muhammad Hafizi Mahmood from UITM, Faculty of Health Sciences, UITM Puncak Alam. Yeah. And sorry, here are our specialty. I myself is forensic entomology. Dr. Jama is a medical microbiologist. Hafizi is medical radiographer. Dr. Lucas Law is a geneticist, and Dr. Sofo is soil chemist. So we make a team so that we can study penguin decomposition from different aspects. So a lot of people ask me, like, how did you go to Antarctica? Is there any flight to go to Antarctica? So just for your information, I was there from 16 December 2019 until 6 January 2020, a total of 3 weeks. And this is how I went to Antarctica. From KL, right, you can take a flight to Dubai, from Dubai to Brazil, and then from Brazil to Chile, Chile to Punta Arenas. Yeah, and then Punta Arenas is a small town at the southern part of Chile. And then from there, you can take a flight to Antarctica. But still, a lot of people think that I actually went to the middle of Antarctica. Antarctica is a huge continent, and I just want to clarify that I did not go to the middle of Antarctica, but instead I just go to the edge of Antarctica, just the peninsula. Yeah, this is what they call the Antarctic Peninsula. So it's the nearest to the South American continent. Yeah. So the station that I attached to is called King Sejong Station. The reason why I choose King Sejong Station is because it is really near to penguin colonies. Yeah. This also makes sense why I did not go to the South Pole, the middle of Antarctica, because there's no penguins. Penguins need the sea, the ocean. Yeah, they need to catch fish. That's why they usually live nearby the sea. And King Sejong Station is a research station under the Korean government, the South Korean government. It's called COPRI. The agency is called COPRI, Korea Antarctic Research Program. For your information, the South Korean government has 2 research stations in Antarctica. One is the first one, the original one is King Sejong Station. I think a few years back, they have the second one, which is called Jangbogo Station. Jangbogo Station is actually nearer to Australia. Let's say you want to go to Jangbogo, then you have to travel to Australia. But because I'm going to King Sejong, that's why I have to go to Chile, and then from there go to King Sejong. And you need collaborators from that station. So we are very lucky. We have 2 collaborators that are willing to work with us. So Dr. Hong as well as Dr. Lee. Dr. Lee is a penguin researcher, and he was also my roommate when I was in Antarctica. And almost every time when we went out to the field, he was my partner. Yeah, we need to go out in a pair, at least in Antarctica, yeah, in a group. Yeah, this is very important for safety reasons. All right, this is King Sejong Station. You can actually go to the website of COPRI, and you can see this is the area map of King Sejong Station. You can see kind of containers on the land, but these containers are made up of the officers, the accommodation, the lab, the canteen, and so on. Later, I will explain to you. I will show you how it looks like inside the research station. So this is what I'm going to do. When I saw a penguin carcass in Antarctica, I plan to take the swab samples from the oral region, the skin, as well as the anus of the penguins. And I will also collect bones from the penguin carcass, right? And second thing I will need to collect is the soil samples. I will collect the soil beneath the carcass and then analyze for the bacteria, nematodes, arthropods, communities. And I will also take a control soil, which is like further away from the penguin carcass. It's just 10 meters away, yeah, from the penguin carcasses. So this is basically the flow chart that I submitted to the grant agency, yeah, Sudan Mission grant. I put up these figures, right, just to make the reviewer understand and easier to digest what I'm going to do. So I'm going to collect bones from penguin carcass, swabs, as well as soil, and all this together is going to contribute to the big data of the necrobiome in Antarctica. So this is my proposal, right? And of course, before you go to Antarctica, you will need to do some preparation. Yeah, you cannot just go. Yeah, you need to do some preparation. And one of the preparation that is required by the COPRI, by the Korean station, is I need to know firefighting, first aid, and sea survival. So I signed up a class, in PJ. This program is actually for offshore engineers, for those engineers who work in the middle of the sea. So actually, it's the same training. So in this picture, that I need to swim out from the helicopter. They put me in the helicopter and sunk to the bottom of the swimming pool, and you need to save yourself, unbuckle yourself, and swim out from the window, something like that. Once you got the certification, then you can make the trip. So this is the day of departure from KL to Chile, and because I'm going to Dubai, stop in Dubai, so I took Emirates. Okay, so after 39 hours of flight, finally I arrived in Punta Arenas. Yeah, just for your information that only myself that able to go to this go to the Antarctica because the grant is only unfortunately only allowed one person, yeah, to travel. Yeah, it's like 150,000 maximum by the grant, so only one person can go. Yeah. So this is Punta Arenas, the city in Chile. I just want to show you a picture so that you get some idea about how this small town looks like. This is the view of Punta Arenas. Pretty much everything is here. Yeah, all right. It's quite a peaceful town. Yeah, you can see some shipwreck near the shore and the bridge. Yeah, and they have a very interesting food called empanada. To me, this is just our Malaysian curry puff. Yeah, although it tastes different. Yeah, but this is something that you can relate to. All right, so those are the luggage that all I have, yeah, that I will bring to Antarctica. One is my backpack, the other one is my laptop bag, and the third one is the cooler box. Of course, now it's empty, and when I bring back the sample to Malaysia, then I'm going to use this cooler box. And this is the hotel that I'm going to meet with those Korean researchers. Yeah, they just asked me to go to this hotel and we will be there waiting for you. I just have this instruction. So on the morning, on the day, I went to this hotel at the lobby, and yeah, many of them, they are ready to go to King Sejong Station. And all those researchers are mostly they are scientists, researchers from Korea, as well as some researcher or university professors from South Korea. They join together and then they go in the team. So I was, just go with them. And once we were in the airport, yeah, this is the Punta Arenas airport. It's a small airport, and you can see this is the place, Antarctica, that you're going to Antarctica. But just for your information, going to Antarctica is not by commercial flight. There's no commercial flight or what we call regular flight. There's no such thing. If you want to go to Antarctica, you really need to book a private jet, a private flight. That's why the flight ticket is very expensive, and because we share the cost with all other researchers. Yeah, but still expensive. Yeah, it's a private jet. And here, this is the private jet that we have that we use to go to Antarctica. Yeah, yeah, with the jet engine. So after one hour and 30 minutes flight, and finally we arrive on the continent of Antarctica, the Antarctic Peninsula, which is the island, right? And this is the airport. And you can see that there is no proper road on the airport. Basically, Antarctica is undeveloped. It's very natural. And this is how the plane just landed on the ground, on the road condition like this. It's actually quite scary, yeah, and quite risky as well. And of course, it totally depends on weather. If good weather, then they will, they will launch the flight. If not, you have to wait until a certain time or suitable time. Sometimes they delay your departure as well. And this is the airport, yeah, very simple, very natural. And yeah, those are the car. Because in the same flight, we have Chinese scientists from China, because the Chinese station is actually nearby Korean station. There are several stations nearby to each other. So sometimes those scientists also join the same flight, and their cars and their staff are already waiting to pick them up. Yeah. So for Korean station, we have to wait. Yeah. And this is quite of an interesting building we saw in Antarctic. My first impression. It is actually a church, and it's built by the Russian station. Yeah, an Orthodox church. I believe this is the first church in Antarctica. And now for Korean station, we have to use a boat, and this boat they call Zodiac. Yeah, they call it Zodiac because we have to sail on the sea for half an hour in order to reach at the Korean station. So you have to suit up with the safety suit. Yeah. Okay. So those are our Korean counterpart. Yeah, they have all the zodiac ready. And this is on the way to the Korean station. As you can see, the landscape is covered by snow and ice, and the seawater, I believe, is cold. Yeah, as you can see, the color, yeah, it's cold. Okay, so when we are approaching nearby, then we can see a ship near the station, and we see some interesting shape iceberg on the sea, and this iceberg looks like a swan. Finally, yeah, we arrive at King Sejong station. As you can see, they actually put out Malaysian flag there. This is actually a courtesy, so a diplomacy, because they know there's one Malaysian is coming, so that's why they put it purposely to welcome the representative. So you feel warm in Antarctica. Yeah, of course, you can see the American flag there because there's an American attached to this station, as well as a Spanish and Argentinian. Yeah, all right. And this is the water resources, the water supply to the station. Yeah, as you can see, they actually melt the snow so that they can use the fresh water they supply to the station. Okay, so now we enter the research station. Yeah, basically it's just like normal, right? It's a corridor, nothing special. Only the address is very different, right? But pretty much pretty normal. Yeah. And this is the canteen. So after that, after we arrive, we gathered here. So the station master is going to brief us about the rules and regulation to stay in the station. Yeah, and most of us actually stay here for about 3 weeks or 1 month. Yeah, but the crew members, they stay for a year in the station. Like the station master, the chief, they actually stay for a year. After a year, they were back to Korea, and another team are coming to replace them. Yeah, and this is the meals that provided, or they have a chef, and they have 3 assistant chefs, yeah, that cook. So you do not need to worry about your food, yeah, because the chef is there, they serve you, they cook, but you have to eat whatever they cook, right? Yeah, so they have no choice. You cannot order from the menu. So they provide your breakfast 7 to 7:30, right? After 7:30, there's no more breakfast. So you have to be on time. Lunch 12 to 1 p.m. and dinner 6 to 7 p.m. Yeah, rice, they have a soup, and they have kimchi. Yeah, kimchi every day, every meal, and I didn't miss any kimchi there. Yeah, almost every meal I have kimchi. So after I came back, until now I actually did not have Korean food. Yeah, because I had enough there. Yeah. So in case you are still hungry after the lunch hour or snack or supper, you're still hungry, there's a 24/7 food supply for you. Yeah, the instant noodle and so on. That's why I have instant noodle. Sometimes I cook. Yeah, and this is the research lab in the station. Yeah. They have almost all the common equipment and materials. But if you need any specialized equipment, then you will need to bring it yourself. Yeah. So I brought over some of my equipments because not every one of them are working on insects. Yeah, all these researchers, they have all different backgrounds. Some study atmosphere, some studies the ocean, some study penguins, fungus, lichen, and everything. So the basic stuff they have, but you need to bring your own if you need any specialized equipment. Yeah. Well, I would say this lab is good. They have a microscope, the group microscope that you can use. They even have a journal club in Antarctica. So my friend was presenting. Because of me and some other foreigners, they have to use English, yeah, so that we can understand their research interests. And they have a lounge. Yeah, after the other lunch or dinner, you can take a rest here in the lounge, and you can play video games because there's a TV there, obviously. Yeah, they have a ping pong table. Yeah, you can play ping pong when you're a bowl. Yeah, and they have study rooms. Yep, when you're not doing research, when you are not in the field, you can work on your publication or your emails, communication, writing, all can be done here. And they have one one clinic. They have one doctor. Yeah, they call it Sejong Clinic. Yeah. Well, I myself fell down 3 times when I was in the field. You know, due to the ground is very slippery. sometimes, you know, accidentally I stepped on the rock and I fell down. I got bushes, so I went to this clinic. Yeah, and they have X room in case we have fracture. Yeah, so they can do there's only one doctor there, and perhaps he can help for some minor surgery. But if you're in very serious medical condition, they have to send you back to Chile, right? And they have one Sejong library. Yeah. Well, the good thing is this station, they have 24/7 Wi-Fi. I think there's one hour break where there's no Wi-Fi because they need to communicate with Korea. But other than that, you have Wi-Fi. The Wi-Fi is not like super fast and it works very well. But you can send some basic WhatsApp messages. I think or Messenger. I think that is good enough. At least you can still connect with your friends and family members. Yeah. They have gym. Yeah. For the staff as well as, can you believe it? They actually have a court, a badminton court, or you can play sepak takraw here as well. This how amazing this Korean station, and they even have a museum just beside the what I call it, the accommodation. Yeah, they have a museum, and this museum is for the visitors like ambassadors, and there are some VIPs. They come to their station to visit, and they will bring them here. Yeah. It looks just nice, very nice. And this is my room, just for your information. Yeah. So inside the room, this is my double decker. Yeah. Basically, only 2 persons in one room. They provide you with chair and table, and there's a window. When you look outside the window, it's beautiful. Yeah, a beautiful view. You can see there's one guy here on your lower right. This is my friend, Professor Moon. This how small of human beings in front of this spectacular great nature. Yeah, we are just very small. We are like ants. Yeah. And sometimes when you look out the windows, you can see ice, floating ice on the sea, and you can see something in the middle. And when you zoom in, it's a leopard seal. Leopard seal, yeah, that hunt for penguins. And because when we went there in Antarctica, it was during summertime, so during summer season. So in summer, the sun never sets. Yeah? You have about 3 months of total sunlight. And I took this picture at 12:03 a.m. And if you have noticed the white group, the white color group, this is actually for telecommunication, for internet and Wi-Fi. So it's very important tools there. And in order for me to get my penguins, I need to walk, okay, from the station to the penguin colony. And it's an area called ASPA 171. ASPA stands for Antarctic Specially Protected Area. It's just like national park. They protect the area because the area is the penguin breeding site. So you need to have permits in order to get into this area. So I applied the permits from Malaysia as well as Korea, and they allow me to go with them. So one way is 2.5 kilometer, return is another 2.5, total 5 kilometers. One I go out, and sometimes I need to go twice in a day, so 10 kilometers per day. So in total 3 weeks, I have walked from the station to the penguin colonies. I think about more than 280 kilometers. That is the total mileage that I have walked. Yeah, so a lot of physical activities, right? And this is ASPA number 171. We reach, and then we need to climb the small hill. Yeah, and it's very difficult to walk because it's covered by snow, and when you step on it, your legs just sink into the snow. So you need to pull it out. So a lot of energy. Yeah, energy is so energy consuming. And we finally arrive at the penguin rookery, and you can see those penguins, they are going uphill, and they create the natural pathway, and this is what we call the penguin highway. Yeah, it's all created by them. Yeah, they go up to the hill, and then they have their nest there, and they when the nest, the purpose of the rookery is to take care of their young, the chicks. Yeah, those are the adult penguins, Gentoo penguins. Yeah, this species is called Gentoo penguins, and those are the Gentoo penguins as well. And on your right hand side, and this is chinstrap penguins. They have 2 species there, 2 different species. Yeah, there are some pictures that I took. Yeah, yeah. This is Gentoo penguins with 2 young chicks. Yeah, right. Yeah, this is Gentoo penguins. And this is chin strap penguins. You see that there's a strap on the near the chin. Yeah, two different species there. And this is a sail. This was sleeping on the snow, and we sometimes we get close to them and we took a picture, right? And this is the way that we go to the penguin colonies, and you can see it's full of gravel, stone, and rocks, and it's not easy to walk on these surfaces, to be honest, and it's so tiring. Sometimes you need to be careful. You need to be careful because some rocks, for example, like this, it just looks like a normal rock, but when you go closer and look at them, it's actually a sail. So you have to be careful. Do not get too close to them, or most importantly, do not step on them. Yeah, because they are wild animal, then they can be dangerous. So you just need to be watchful to your environment. And if you allow me, I would like to share with you some graphic images. This is about my study because I'm looking for the penguin dead bodies. Yeah, so I'm going to show you some carcasses, right? For example, this one, yeah, is a dry penguin carcass. So when I see this carcass, I'm going to take the swab, the soil, as well as the bones, to examine what kind of bacteria are on them. Yeah, and for information, in Antarctica, there is no flies, there is no ants. This is very different from tropics and temperate countries. So how do they decompose? Yeah, so it primarily, I think, is due to the bacteria. That's why we need to take the swab and soil, right? And this again, a dry and remain stage of another penguins. And this is a freshly killed adult penguins. As you can see, there is a big wound on the body, and probably it was attacked by a seal in the sea or somehow, and then the body was washed off on the ground. And this is a penguin chick, the young baby penguins. It was, can you see the punctured hole in the middle? It was predated by a predators. It's a seabird. We call them squaw. Yeah? It's a brown color seabird, and those birds they actually predate on young penguins. Yeah, this one. This is the bird. We call them squaw. And this squaw, sometimes they also pick up the penguin eggs and finish it up. So this is what I do when I saw the penguins. Take the bones, take the swab on the skin, in the mouth part, and as well as the control soil. Yeah, the soil beneath as well as the control. Yeah, to compare the soil chemistry beneath. Sometimes we also saw the bones of the whale. Those are whale bones. That was found on the soil, right? And this is a seal carcass. Yeah. Not only penguins, but sometimes we also got this. Yeah. And this looks like a human dead body. Yeah, to me, at the first sight, from the beginning, it looks like 2 legs there, but actually this is a seal carcass. Yeah. Still, luckily, there is no murder case in Antarctica, so the scientists they are in good working relationship, so there's nothing to worry about. Yeah, and sometimes we see a lot of these insects. Those are insects, the black color one, those tiny one. Those are Collembola. I collected some of them and I brought back to the lab, and they look like this. Yeah, the black color one. Yeah, there's so many. I would say they are like the ants of Antarctica. And we also saw something like this. Yeah, maybe it looks like a soil particle to you, yeah, that attach on the rock. But actually, under the microscope, there are some organisms there. I'm going to show you the image. Just like this. It looks like very normal, pretty normal on this rock surface. But if you zoom in the middle, there are mites. So the organisms are everywhere. Yeah? The living organisms are everywhere in this frozen land. Right. Those are the equipment that I brought to Antarctica, Belize funnels, as well as the Herman apparatus, yeah, to isolate the worms as well as the arthropods from the soil. So when you examine under the microscope, you can see the mites as well as the tardigrades, the water bear, the worms, right? And those are the decomposer of penguin carcasses, I believe. Yeah. And of course, we work in a team, and those are our team members, yeah, the Korean team members. Yep. So we always work in a team, and sometimes there are some international researchers also attached to King Sejong, like this picture on the left. It's the American and Argentinian, and then there's one guy on my right. That guy is a Korean entomologist. So we met two entomologists there, so it's good news. And sometimes when we work in the teams, sometimes you just need to help your friends to catch the penguins. This is Dr. Lee who need to put a GPS tracker on the back of the penguin. So sometimes you just need to help them, yeah, because they are actually short of manpower as well, yeah. So I have a chance to hold the penguins, and this is the GPS on the back of the penguin. Dr. Lee wants to track the movement of the animal, so it's very interesting. And just also interestingly, because we have this welcome party during the at the King Sejong station, so I think it's very interesting experience to share with you. And then we also went through the Christmas, so they have Christmas party, yeah. And this is the chief station, Dr. Hong, on my left, and as well as New Year party 2020, yeah. So they're also like human beings, right? Although we are in Antarctica, but we are humans, so they have some celebration in this lonely land. Yeah. Sometimes you just need to be creative. I found an ice chair, and it's such a spectacular view, right? So this is the whole the overview of the station, and yeah, we need to continuously put Malaysian flag in Antarctica. So we need to take this picture as well as to bring UITM in the world map. And then we give some souvenir to the station chief, Dr. Hong, as a souvenir. And this is the last day of our stay, and we have a group picture, and then after that we will depart back to Chile. And so those are the samples that I'm going to bring back to Malaysia, and I put in my cooler box. And in order to maintain the temperature, I put 3 kilograms of dry ice in the cooler box so that it can, keep the temperature down until my destination in KL, right? Okay, so this is the day that we went back home, well, or so-called went back to the civilization. Yeah, they make joke on that. Return flight, yeah, this is a return flight. So after 42 hours flight, and we have some dramas, just to let you know, it's not easy to bring back the cooler box with sample. So the first drama was my cooler box was missing at Santiago Airport. I was so sweaty, so nervous, because you know what, if my cooler box went missing, it means that all the effort, all the money spent, just wasted, just gone. So we should not, yeah. Okay, and then number 2, inspection of cooler box by immigration officer at Brazil airport. Yeah, they stopped me, they sent me to inspection office, they asked me a lot of questions. Very luckily, at the end, they released me. And you're just concerned, worrying that your cooler box been missing all the way from Brazil to Malaysia, right, in the flight. We heard about lost luggage. So finally arrived in Malaysia with the Antarctic sample on 11th January 2020. And yeah, as the Dr. Jama was there, high enough to pick me up and send the specimen back to the lab. Yeah, back to the lab in the UITM Sungai Buloh. And this is where we kept our sample at the Institute for Medical Molecular Biotechnology. Yep. So feel free to visit. And this is where we in order to store the Antarctic sample, we need to have this specialized signboard. Yeah, and this signboard has to be approved by the Ministry of Agriculture. And after that, the officers and staff from Ministry of Agriculture, they do come and visit us and check on our sample to make sure that we really abide to their law and regulation. Yeah, because those are foreign soils and samples, so it's considered biohazards. So they need to do their job. They did their job well. And also our press conference was also in media like New Straits Times, The Star, as well as the Sina Halian and Bernama as well. And as well as Dewan Cosmic, to encourage and to inspire the generation. So a lot of people ask, what are the impacts to the world with this kind of research? Of course, this is our science, and our science is to understand effective nutrient cycle. Perhaps one day we can actually cultivate crop plantation on the icy land if we understand how the nutrient cycle works, right? Yeah, and necrobiome also useful in forensic purpose, archaeological purpose. Then we can understand how the animal migration, geology, how they adapt to the extreme environment. And this is our contribution. It's coming. Well, we are still working on the research to the world. What are the impacts to Malaysia? I'm sure a lot of people will ask this question as well. So first, you need to raise awareness about the Earth ecosystem to our fellow Malaysians, about climate change, about global warming. And this is true. I have seen with my eye. This is true. The glacier, we are losing out them. They are decreasing in their volume, and this is true. Yeah. Second is to develop Malaysia's scientific capacity and also in our education system, yeah, for our future. And also continuously putting Malaysia on the world map, including Antarctica. But why? Why is it important to put Malaysia there? Yeah? Of course, to sustain a Malaysia presence in polar research and strengthen our research capacity, yeah, in the global platform. Yeah, just for your information, Malaysia is a member of Antarctic Treaty System, yeah, and as well as Scientific Committee on Antarctic Research, SCAR, right? And Malaysian scientists, I mean, before this, there are many Malaysian scientists have been there. They are actively involved and share their findings and data to SCAR, to this scientific committee, and then therefore Malaysia has been a part in the policy and decision making at the international level. So if you are interested to obtain this grant is open every year. Just go to this website, yeah, Sultan Nizam Antarctic Research Foundation. Just type this, Google this, and you can get the form, yeah, Form A, Form B, and the guidelines. Everything is there, right? Those are the priority areas: science and policy research on biology, ecology, geography, everything, yeah, ocean, atmosphere. For 3 years, the grant is for 3 years, and for maximum 150,000 ringgit, right? And because I'm from medical faculty, so those are the topics that we can do. This is what we call polar medicine. You can do, circadian rhythm, electromagnetism, vitamin D, bone density, and so on, right? And even there's a paper published in New England Journal of Medicine. They found brain changes in response to long Antarctic expedition. You can even study the subject, the researcher who stay in the station for a year. Yeah, you can study them as well as their subject. So those are the eligibility criteria for you in order to apply the grant. First, it must be scientific research. You must have a PhD, 3 years experience, and you are a researcher from this university, UITM, USM, and so on. Yeah, only these universities are eligible to apply, right? And you need at least two local or international universities to join you as a team. And in my proposal, I have Dr. Siti Sofo from UMT, as well as Dr. Lucas from So I have 2 local universities. And just need to fill up Form A and Form B. And the deadline of application, of course, is already due, but please apply next year. Yeah, normally early next year it will open, and then the deadline usually in March. So you just send in. So you have plenty of time now to think about the research proposal and form your team. And next year, January, hopefully they will accept, they will open. But need to check again, because now it's under COVID-19 pandemic, whether they allow travel to Antarctica and so on. So far, Antarctica has 0 cases. It's the most safest place on Earth from COVID-19. So the future, now I have a PhD student who is currently working on the Antarctic sample, No Adila Zaini, on this topic. So she just started, this is her first semester. So we are looking forward to the results from her study. The future is from my lab. We are going to focus on this area. And I would like to acknowledge, there's a law agency, a lot of people, COPRI, the YPASM, UITM, I perform, Faculty of Medicine, IAMB, as well as the Jabatan Pertanian and Jabatan Pertanian Veterina. With all these people support, then only we have a chance to go to the Antarctica. So with that, I would like to thank for your attention, and feel free to ask me any question. I hope I can answer this. Right? Thank you so much.
Thank you very much, Dr. Hio. It's always interesting to listen to your talk.
Thank you.
And yes, the floor is open now for anybody who would like to ask a question.
Hello, good morning.
Hi, morning.
Dr. Hio, congratulations for this excellent presentation. Very informative.
Thank you.
Yeah, I have a question. Okay, you were studying about the entomology, yeah?
Right.
What I'd like to know is what are the potential topics that perhaps UITM researchers can embark on? I mean, based on your suggestions to form a group and then to forward the proposal to the Yayasan Antarctica. based on your observations when you were there, knowing that we have a quite comprehensive research area, I mean, researchers, you might want to, suggest what are the potential topic, perhaps. Thank you.
Right, all right. Thanks, Prof. Najib. Thank you very much for your invitation for me to share my experience as well. So it's a very good question. Actually, from my experience, there are many areas that we can venture into. Like just now I mentioned about the polar medicine, which is we can work with Faculty of Medicine, as well as the chemistry, the ice chemistry, the quality, air quality, pollution, global warming, climate change, and in fact, before you apply to this proposal, YPASM, they have a guideline, what kind of the questions that are internationally needed. Yeah, I believe they have 88 questions that are needed to answer for the Antarctica problems. So just now I mentioned about the climate change, global warming, those are the priority research area. And as well as the conservation, biodiversity, yes, and those are very much encouraged to apply. And of course, when you have a title and your scope, you will need to find out which research station is the most suitable for you, which research station to go. If you need a penguin, then you have to find a research station that's near to the penguin colonies. And then from there, once you have targeted the research station, then you start contacting the scientists working in the station, and hopefully they are interested in your proposal and willing to work with you. So there are several steps there, yeah, but it can be done. It can be done. So now the most important now is just look to the website and look for the 88 questions of Antarctica. What are the priority research questions that need to be answered? And then you form up your team. After you form up your team with all the expertise, draft the proposal, then target for the research station. After target the research station, contact the researcher. Yeah, so those are what I did for the past 1 year in order to be.
Thank you, Dr. Theo and Prof. Najib. Dr. Theo, we have one more question from the chat
Yes.
from Prof. Madia, Dr. Angeline, if I'm not mistaken. She mentioned, thank you for your interesting expose on the penguins in Antarctica. I'm an arts person. How can I be part of this scientific research? I'm very interested in the sort of work, but it certainly is not my area. How can I, and people like me not in the field, offer our contribution in this sort of work? Thank you.
Right. So yeah, it's a very good question. Thank you for the question. It depends on your study area, because they are researchers, they are working in the station. So we are human beings. So if you are from the art and social sciences, perhaps you can do some psychological evaluation, that kind of an organization study, how human beings adapted to that extreme environment. That is one. Number two is sometimes when we talk about polar region, it's not necessarily Antarctica. There's also Arctic region. Arctic is also very isolated. But the good thing about Arctic is they have their natural human population there, yeah, the indigenous population. While in Antarctica, there's no human beings there. Yeah? All human beings are introduced, are imported to Antarctica. But in Arctic, you have natural population. Then you can do some social study about their lifestyle, their food, their diet, perhaps their religion. There are many things to study. I hope I answered this question.
We still have Dr. Q around. Anyone would like to ask him any questions? Please do so. I have a few more here that really like your presentation, and some even asking for your slides and your email address.
Oh, so sure.
Yeah. If you may share your email address maybe, and I think this session is recorded and then later will be available from the RMC, is it, if I'm not mistaken? So later can be available to everybody.
Sure. Yeah, I just put my email address there.
Yeah, thank you so much. Another one is we have a question. is from Prof. Madia, Dr. Sadia Sultan, referring to you. Doctor, I'm working on the bioinformation using microbes. I have worked on few, and one of them showed interesting results. I need your email to ask for the details. So we have already provided your email over there for them to contact you.
Yes, bioinformatics, bioinformation, and those are very it's trend now, very hot topic. And while I was there, the Korean scientists are working on the microbiome of the researchers' intestinal tract of their own researcher. Yeah, they are working on how the microbiome changed from the beginning when they just first came from Korea, and maybe in the middle and after 1 year. How the microbiome change. I think this is a very interesting study. So for sure, sequencing, bioinformatics is the way to go.
Okay, yeah, that's true. Yes, very much. These are the in-thing studies, and we should be looking forward into. And another question from Dr. Amirul, who is asking, interesting sharing session. How many social scientists were there, if any, if you came across anyone?
Yeah, yeah, it's a good question by Dr. Amirul. To be honest with you, I haven't seen any social scientists while I was there for the 3 weeks. I'm sure maybe other seasons there are some other social scientists coming in, but when I was there, I didn't know any of them. I know there are some scientists they are doing some mapping of the area, but well, I believe that is a geography, not really the social aspect of it. Yeah, so hopefully, Dr. Amiro, you can put in the social proposal. Hopefully, you can start your social study in polar region.
Okay, we have one more question here from Zano Ghazali. Thank you, Dr. Hyo. Good sharing. Just out of curiosity about the sampling, are you bringing back the bones solely, which part, or do you sample extraction first?
I bring back the swab, bones, and soil. 3 types of samples. Yeah. So my role was as a sample collector. Yeah.
Yes.
So I was there to collect samples, and then I brought here. Everything started in Malaysia.
Of course, Dr. Yu, maybe you can mention about every part that you need individual permit to bring back to Malaysia.
Sorry?
So every sample that you have collected, individual samples, the soil, the bones, and all that needed individual permits.
Yes, a very important point here, Jama, you just reminded me. We need import permit from Malaysian government as well as export permit from the Chilean government, because you are bring out sample from Antarctica. Okay, here is the thing. Antarctica belongs to no country. There's no country of Antarctica. So the Chilean government, if you go through the Chile, then you have to apply permit from the Chilean government. But I was very fortunate. The Korean station actually helped me to get a permit. For the import permit from Malaysian government, you have to work it now before your departure. You have to get all the process done. Then only you can bring back the sample. Once you arrive in KLIA, you need to declare to the officials that this is the permit and these are my samples. They will check and they will take picture and something like that.
Yes. And we have our YPSM representative here as well, mentioning that intern Fazlin is with us. And he says, since UITM is renewing the MOU with them for the next 5 years, so everybody welcome to apply for the next cycle. And he's put the link over here. So for the people who are interested, they might look into that and explore further. I think the safest place not to do research probably is Antarctica, free of COVID, and you can do your work free in peace of mind. Do we have any last burning question for Dr. Hyo?
Hello, Dr. Jama.
Yes, Prof.
Hi, Assalamualaikum. Very good morning. Congratulations, Dr. Hyo, for a very exciting presentation, and I think it's new opportunities for UITM, certainly, and perhaps with collaboration with other universities locally, internationally. Just two quick questions, Dr. Hio. Firstly is, do you foresee any possible niche areas, research niche areas that that is within the list that you mentioned that UITM could possibly champion? Number one. Number two is, although there is no local population that are group of people who are staying permanently in Antarctic, but has do we know what sort of how big is the population? Is that just the researchers or who else is staying there?
Yeah.
Just wondering whether there have been any studies looking at the health status of those people who reside in the Antarctica, including the coronary risk factors.
Right, Prof. Thank you very much, Prof, for your question. I would like to answer the second question first. Because Antarctica is extremely cold during the winter season, which is 6 months long, that time, not many researchers were there, right? Most probably only the crew members that take care of the station for the whole year. Like they are like 20 or 30 people. They are not many, right? So I think during summer, that is the most active season, which is from December to February. Yeah, these 3 months is the golden season for researchers to go to Antarctica. All right. If you are the wildlife ecologist or biologist, that is the best time because a lot of wildlife are active during that time. You can observe them. Unless your study niche area is about electromagnetism, you want to determine the aurora or something, then probably you might need to go during winter season. So the best time and the most active time is summer. And there are more people there. I would say more than 1,000. Even in one American station, McMurdo station, is like a village. I heard it's like a village. It's more than 1,000 scientists there, and they have a restaurant, cafe, hospital in Antarctica. So you can imagine how big it is. Back to your first question, Prof. I think we have many, as you know, we have many disciplines in UITM that we have all full potential to thrive. But for now, I think I'm the second person from UITM that went to Antarctica, and so I'm kind of the biology, ecology, or medicine. But the first person who went there is Associate Professor Dr. Huzaini, who is from engineering. So I think he's studying the magnetism and atmosphere, if I'm not mistaken. So I think this group, engineering and medicine, for now, we already have some experience there. But of course, we would like to see more sciences or even social sciences to be our representative in Antarctica as well in future.
Thank you. Thank you very much.
Okay, thank you very much, Prof. Hafiza and Dr. Hio. Any last questions before we wrap our session?
Yeah, I would like to say, good to see Intan here, the YTASM representative here. Yeah, Intan is very, very helpful, especially when you need to apply for the permits, the import permits. So I would like to say thank you to Intan for all the hard work and all the communication done. Yeah.
Indeed, yes. We just research in this magnitude. We need a lot of help. We need a lot of collaboration from everyone. And I think without that, this type of work's not possible. So I hope a lot of people are thankful to you, Dr. Hio, for the sharing session and very informative. Everybody, I think, are motivated to explore beyond the areas or not only the four walls or beyond the comforts of the places that we are. So indeed, very interesting, as I said, as always, and it always gets me, from the day we started talking about this project until today, it's always been very, very interesting.
Yeah.
Yes.
we start from a very humble beginning, Dr. Jama. If you might remember, yeah, with Mr. Hafizi, we were in your office. Three of us were in your office. We're talking about the proposal, how to study and what to study from a very humble beginning, and we actually never expect that we will actually get it right. Yeah,
very
we're actually just trying our luck. That's all.
Exactly.
Yeah.
We always have to try. We will never know.
Exactly.
I think we had over 230 audience today listening to your talk. It's very fruitful and interesting. Okay, yeah, many people thanking you for the presentation and sharing session. So thank you to the TNC RMC and Dr. Hyo Chong Chin and the organizer for this wonderful session. With that, we end this session. I'm not sure about the availability of the slides or the video. That one we get back to the RMC team.
Yep.
Okay, that's all from me and the presenter, Dr. Hyo. Thank you very much, everybody. Assalamu alaikum and have a very good day.
Thank you. Good having you all. Thank you.
Thank you.
Thanks.